comes the third law of third law of genetics or inheritance that we call as is the law of independent assortment. So it states that the completely different pairs of alleles are passed on to the offspring independently of each other, that is during gamete formation two genes segregate independently of each other without influencing each other as well as of the other trait, and the inheritance of one gene does not affect the inheritance of other gene, they are not related to each other. And when the cell division happens to create the to form the gametes, there is no relationship between these two genes that one is influencing the other one. So, that does not happen. We have seen that there is two types of gene, that is recessive gene and a dominant gene, but there are some cases when the experiments of were conducted for the length of pea plant as a trait, the phenotypes of the hybrid or the resulting plants resembled either of the parents. However, when experiments on pea were reported using other traits, some of the other traits were studied in other plants also, it was found that sometimes F1 generation had a phenotype that was not resembling to either of the two parents, which was actually in between the two parents. So, the experiments that were conducted to confirm this incomplete dominance or inherited intermediate inheritance was done on the flower color in dog flower, that is snapdragon of Antirrhinium species. So, this was done to understand that what is happening when how a pink color flower is created when a cross pollination was done. So, in this case true breeding red flower and true breeding white flowered plants in dog flower was done, and it was observed that F1 generation was pink, and then when F1 was self pollinated, the F2 resulted in a ratio which was different from earlier one, from 3:1, now it is 1:2:1, where 2 were pink, 1 was pure red and 1 was pure white. The hybrid plant, the hybrid alleles have shown a different kind of color which was not present in the parent color. So, the genotypic ratio is exactly as proposed by Mendelian monohybrid cross, but phenotype ratio had changed from 3:1 to dominant is to recessive ratio, as R was not completely dominant over small r. So, this was a different thing that was observed, where the genotype was exactly the same, that one would be pure dominant, one would be pure recessive and 2 would be the hybrid one. But the phenotype actually changed. In the earlier experiments, what we have seen in the pea plant, it was observed that phenotype and the genotype ratio remain the same, where it was 3:1, but now the situation has been changed. Now, the phenotype has been changed as 1:2:1. So, this is an example of incomplete or incomplete dominance or intermediate inheritance. So, that was a different concept from Mendelian's experiment, but this was again related to the fact that it is associated with the genes and their alleles. The only thing that was that there was no pure dominance of one allele over the another one. So, there was a different concept that also came besides incomplete dominance or full dominance. The third concept that we can see was co-dominance. So, till now we have seen either it is completely showing or it is partially showing. Now, we are looking that what is the stage of dominance where the both are equally dominant. So, the case where F1 generation resembles both the parents in a heterozygous organism, both the alleles express themselves completely. It is the concept which is different from what we have seen so far. Now, both the alleles are expressing themselves fully. So, we will try to understand what is the case when heterozygous organisms is there and what is the stage of the allele and how it is expressed and what different kind of things we can see. And this is the co-dominance we can observe, and the best example to study the co-dominance is the different types of red blood cells that determine the ABO blood groups in human beings. So, we all have heard that everybody has different blood groups, and B positive is the most, B is the most common blood group that we all have, but we will try to see that how B is there, what are the alleles, what are the genes that are associated with it, how it is impacting, what is the stage of dominance, which gene is more expressing, which is less expressing, and what are the reasons behind that. So, codominance. So, ABO blood groupings are controlled by gene I located on chromosome 9. So, this is the fixed location. You all must be aware that there are DNA double helix structure where different kind of genes are located, and genes are there to carry on the heredity, the different traits, and the running of the cell also. So, they have all the information located within themselves. So, this is our blood group is located at gene I on chromosome 9. So, this is a very frequent question that occurs in NEET as well. So, at which gene and what chromosome is associated with our blood groupings. So, the plasma membrane of the RBCs, that is the red blood cells, has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. So, which kind of sugar polymer would be present on RBC would tell us our blood group. So, the plasma membranes, and these sugars are present on the plasma membranes of RBCs, and everybody's body is different in terms of blood groups, because the sugar polymers that are present on their RBCs would be different, and this is what we receive from our parents. So, the gene I has 3 alleles, that is IA, IB and small I. So, this is how we present the alleles of the blood groups that are associated with human beings. So, the allele IA and IB produce a slightly different form of sugar, while allele small I does not produce any sugar. So, two kind of sugars are present on RBCs which guide the final blood groups into our body. So, one is IA, another is IB, because small I does not has any sugar, so it is not impacting our blood group. This is type of allele that is present in our genes, which will finally tell us what blood group we would be having. So, as humans are diploid organisms that we discussed, each person possesses any 3 of I gene alleles. It can be IA, IB or small I, and the dominance of these 3 different kind of alleles would guide the presence of the blood group into our body. So, IA and IB are completely dominant over small I. When IA and small I are present, only IA expresses. When IB and I are present, only IB expresses, because these two are dominant genes, where small I is a recessive one. However, when IA and IB are present, when both of these are present, what will happen, because both these are dominant one. When both these are present together, they both express their own types of sugars because of the codominance. Here comes the concept of codominance. When both the dominant genes are present, what is the situation? In such cases, both express their own type of sugar. So, when they both combine, then the RBCs would have both kind of sugars which are protruding from the RBC surface or the cell surface. Since there are 3 different alleles, there are 6 different combinations of these 3 alleles that are possible. A total of 6 different genotypes of human ABO blood types are present. So, we all have to understand that there are different kinds of blood groups that we know as A, AB, B and O. So, that is, these 3 different alleles are responsible for different kind of blood groups that are present from different combinations, that is the 6 different combinations of these 3 alleles. They are possible, creating a total of 6 different genotypes. Here we are talking about genotypic situation, because we have 6 different combinations possible from these 3 different alleles. So, the genetic basis of blood groups in humans, there are 4 phenotypes and 6 genotypes that are possible. If we see from this particular table, when IA and IA from the gametes are combining together, then the combination would produce IA and the blood group of the resulting progeny would be A. When the situation happens that IA combines with IB, then because both IA and IB are dominant, they would express themselves completely and the progeny that would be created would have a blood group of AB. When the gamete from IA combines with progeny with gamete of small I, then the resulting situation would be that in this case only IA is dominant and would be expressing. So, the blood group of the progeny would be A. And if again the gamete from IB combines with progeny of IA, the progeny that would be created would, because of the codominance of both these, the progeny would have the blood group of AB. When the gamete from IB combines with the progeny of gamete of small I, then the progeny would have, because only B is dominating, then the blood group would have been B. And then when the small I combines with small I, then because both are recessive genes and they do not have any kind of sugar on them, the blood group would be O. So, this is how the different blood groups are decided by the genes through the combination of different kind of alleles and the presence or absence of sugar on the red blood cells. So, this is a very interesting concept that which blood group would be generated. We can understand how a blood group testing of the parents is done to understand what kind of blood group would be for the progeny. So, this is how it can be tested, because the combination of different alleles would produce different kind of blood groups among the progeny. And this is a very interesting Punnett square or representation of the different blood groups and the combination of different alleles, where we can understand if IA and IB or the gametes from IA and IB parents combine, then the blood group would be AB. And I mean, there is no other possibility, then blood groups can be changed. I mean, if IB and IB gametes are combining together, then there is no possibility that the offspring would have O blood group. So, that is not the case, because they are all associated with the genes and the alleles associated with them, which are producing different kind of sugars on them, and the blood groups would be fixed depending on the combination of the alleles that they are getting from their parents. So, the offsprings would have these combinations. It is that case when the parents is heterogeneous, then we can also predict that all the 4 combinations would be possible in the progeny. But when gametes have created and they have combined together, then the blood group cannot be changed. If IB and IB are joining together, then only B blood group is possible. AB or A blood group won't be possible. So, it is the combination of the 2 gametes which are of different allele combinations, because we have 3 different kind of alleles creating 6 different combinations, would depend on the combination of the gametes, resulting in the formation of sugars, and in advanced stage the formation of the blood group of the progeny. So, there are 4 phenotypes, as we can see. One is A, another is AB, then the third one is B, and the fourth one is O, from the 6 genotypic combinations that are possible. So, examples, this is a very good example of multiple alleles for one particular trait. So, we do not have 2 alleles now, we have 3 different alleles for one particular trait, that is IA, IB and small I. These are 3 different alleles, all are responsible for the blood groupings in the human beings. So, there are more than 2 alleles governing the same character present at the same locus of the chromosome, that is gene I of chromosome 9. All the 3 alleles would be present on that particular chromosome. So, in summary, the inheritance of blood groups in humans show dominance, codominance and multiple allele. So, dominance is when IA and small I are present, it is showing the dominance where IA is dominant over small I allele. So, the blood group would be A. So, codominance is an example we can see here that when IA and IB are present together, they both would be dominating and would be producing the blood group of AB and multiple allele. One particular character, that is the blood grouping in human being, is associated or is guided by 3 different types of alleles, that is IA, IB and small I. So, it is the thing where we can see all the different concepts within one particular concept, that how blood groupings in humans are guided by. So, from this lecture we have all understood that what are the different laws which guide the inheritance, how inheritance is guided through different kind of genes, because earlier that were known by Mendel as factors, but actually they are genes, which was the term was not have been not introduced at the time of Mendel. We have seen 3 different laws from Mendel, that is law of dominance, law of independent assortment and law of segregation. These the law of dominance shows that the first progeny, even if it is a hybrid, then also the dominant gene trait would be expressed in terms of phenotype, although genotype makeup would be the hybrid one, but still the expression or the phenotypic expression of that particular gene is based on the dominance of any particular allele on that gene. So, that was we have seen in F1 generation and F2 generation also, where heterozygous conditions it was justifying 3:1 ratio. Then came the law of segregation. I am reiterating these laws because these laws are really important to understand the different concepts that we would be discussing further in terms of diseases, sex determination and all those concepts we would be discussing in coming lectures. So, the law of segregation, we need to understand that for any trait, each pair of allele of a gene segregation and one gene passes from each parent to an offspring. It is not that half of the genes have been passing from one generation or one parent to another generation. All the genes, irrespective of each other, would be passing from each parent to the offspring. And these two alleles do not mix when they come together. If we can see one time more, we can see the picture of the allele. So, this is an allele. If we can see at the last at the bottom picture, they are on separate locations. They can't be mixed together. So, one gene on one allele on one gene and another allele on another gene. So, when it is passing, they are not mixing with each other, that a in between situation occurs. No, it is like one allele would be passed from one parent to another progeny, irrespective of effect of other or irrespective of its any kind of mixing. It would be a pure transfer. So, this is how the law of segregation comes into picture, where there is no intermixing or no chances of passing half of the allele to another gamete or the offspring. So, they do not mix when they come together in a hybrid pair. Also, when they come together, then also there is no mixing. When they are passing from one generation to another, there is no mixing, and when they pass on to the next generation, then also there is no mixing when they come together. They come together independently of each other. Then they do not show any kind of blending in F2 generation also. And do remember that a homozygous parent produces all the gametes that are similar. If we consider TT example, capital T, then the gametes that would be produces with one with capital T and other also with capital T. So, they would be similar in all the aspects. While in case of a heterozygous situation, for example, capital T and small t, when the gametes would be produced, one would be capital T and another would be small t. So, two different kind of gametes would be produced, each having one allele But and again you have to remember that they would be in equal proportion. It's not that half of allele is transferring from one generation to another generation. It would be that one complete allele would be going to one gamete and another allele would be going to another gamete in equal proportion. There can't be half or in between situation. Expression of allele, we have seen that incomplete dominance that we have seen, but still the passing of allele is complete. That is, the expression of allele can be modified or has been changed in some kind of traits. We have seen that there is incomplete dominance, but the inheritance was complete. So, there are two different aspects to this, that inheritance was complete of those alleles, but yes, the expression of those alleles was different, leading to incomplete dominance, because we have seen two different kind of traits or two different kind of expression of those genes. One is those alleles, one is dominant allele gene and another is recessive gene. But when a in between situation occurs, then it is known as incomplete dominance. But we have law of independent assortment, where completely different pairs of alleles are passed on to the offspring independent of each other. Again, there is no influence of one gene on the other or one allele on the other, that okay, no, half of it should pass or more of it should be passed to one gamete and less to be to the another. No, there is no such thing. Complete different pairs of alleles are passed on to offspring independent to each other, and two genes segregate independently, and so the traits also. So, the inheritance of one gene does not affect the inheritance of any other gene. For example, if we discuss about the gametes, when gametes are produced from one parent, two different kind of gametes would be produced if the parent is heterozygous, and similar kind of gametes would be produced if the parent is homozygous. But the gene content in both these gametes would be exactly the same. The percentage of the gene content that is passing from one parent to another would be exactly half of what the parent had, because we are diploid, the parent had two. That is, if we see for an example of a chromosome, 23 pair of chromosomes are present in the parent cell, and when the cell divides into two to produce different gametes, then 23 chromosomes would pass from to one gamete and 23 would pass to another gamete, but the content would be the same, and one kind of gene which is passing from one parent to gamete would not influence the amount or the number of chromosomes that are passing to another gamete. So, they are not causing any kind of hindrance or affecting the inheritance of another gene. So, it would be exactly the same, the amount and the content would be the same. The allele which is transferring from the parent to the next generation would be different, but then again if it is a heterozygous parent. If it is a homozygous, then allele also would be the same as we have seen. So, in terms of codominance that we have seen, ABO blood grouping is the best example to study the codominance, where two different kinds of alleles are expressing themselves, and not only expressing, they are expressing them equally, fully. They are not that if A and B are present, A is expressing more and B is expressing little less. No, it is exactly the same. What A is expressing would be the form of what B is expressing, and it is present because we need to understand that where our blood group dominance is located. It is located at gene I on chromosome 9 on RBCs, and what is blood group, we need to understand that also, that how blood group is what is defining the blood group is the type of sugar that is present on the RBCs. That is also very important to understand, that it is nothing else but only the type of sugar that is present on our RBCs. Sugar is basically CHO combination, carbon hydrogen oxygen combination, which is producing different kind of chain lengths and how long the sugar would be. So, that is the type of sugar present on our blood cells, RBCs, red blood cells would define what kind of blood groups we are having, and 3 different alleles present in our body would define 6 different genotypic combinations for the production of blood groups, and there are 4 kind of blood groups that can be possible from this 6 combinations that we have seen by producing the different combinations of IA, IB and small I. This is how our blood groups are created from for 4 different phenotypes from 6 different genotypic combinations that are possible. But we all have to remember that all different alleles are present on the same locus of a chromosome. If we have seen that gene I is guiding on chromosome 9 is guiding our blood groups in for IA, then for IB also it would be gene I at locus at chromosome 9 would be guiding the blood group in another cell or another person. So, the location, the locus on the chromosome for a particular allele would be same. It is not that IA is on a different locus and IB on different locus and small I on different locus. It is only the allele who is the way that is expressing is different, locus would remain the same, and it may be a 2 allele trait or a 3 allele trait. We have seen in humans it is a 3 the blood groups is 3 allele trait, where the 3 different kind of alleles are present, whereas in case of pea plant we have seen the maximum traits that were seen was 2 allele trait. The stem cells was represented as TT because it was a 2 allele trait. Then for different color of the seed it was YY that was seen. For seed shape, that is wrinkle or a round shape, it was again a 2 allele shape, that was RR. So, it all depends on that how many alleles are present guiding that particular trait. For blood group it is 3 trait mechanism. So, from for blood groups we have seen all the 3 things that we have studied so far in this introduction to hereditary chapter, that it is showing dominance, where if IA and small I are present together, then only IA will express because it is a dominant gene. We have seen codominance when IB and IA both are present, both with dominant equally and produce the AB blood group, and also the multiple allele that I have just highlighted, that in humans we have 3 different alleles guiding the blood groupings, that is IA, IB and small I. So, this is all about the introduction to heredity. We have seen what is the difference between gene and allele. So, how these things would take care of different traits in terms of genotypes and phenotypes. Again, I am highlighting the point that phenotypic expression would not tell you about the genotypic makeup of that particular trait. So, we need to study the genotype to get the exact genotypic makeup of that particular organism. So, phenotype may be different from the genotypic ratio because of the presence of dominance, codominance or incomplete dominance. Thank you.